Alrighty, let's go on to the variations on Mendel, which is some of the non-Mendelian genetics, it's also called, when things aren't simple, dominant, and recessive. We're going to talk about 5 different types here. Incomplete dominance, and let's go through these quickly because we'll see all of these. And pleiotropy is in the book also, but we're going to skip that one. So let's start. Incomplete dominance. Incomplete dominance is like it sounds. One trait is not completely dominant over the other. One doesn't just hide. It's actually, it looks like to be a blending. It's a blending of traits, not necessarily a blending of alleles. So the F1 generation will show a trait that's in between. You have red and white will make a pink offspring. Snapdragons, the flowers, there we go. You cross red and white snapdragons, and your next generation is all pink. There's no red, there's no white. Why is that? Because there's been a blending of the appearance of the traits, not the alleles, just the appearance. And the F2 generation, so you take those F1 generations and breed those with each other, and you're going to end up with one red, the dominant, one red, you're going to have 2 pinks, and then 1 white as well. So it's like the red and the white reappeared, and they're still pink. When we do Punnett squares, we'll get some mix of those. So here you see what's going on. You start off with red, and notice how that is, excuse me, how that's identified. C big R, C big W, color red, color white. The offspring, look at this as an RW. There's a red and a white, it's going to have the appearance of pink. And then as these separate out, law of segregation, we're going to have CR and CW. So this will be here, and this allele comes over here. Same way for the other side. CR will go there. And with the Punnett squares, look left, look above, and you'll have, without my lovely picture there, you'll have 2 pinks, 1 red, and 1 white. And you can see that double there. You can check on your own with your own copies of the PowerPoint. Codominance is like co-captains, 2 equally dominant. They are both going to be expressed. One doesn't fight the other, one's not above the other. They're both equal. The text uses the blood grouping. Another type of blood group besides our ABO blood groups would be blood type M, blood type N, and right in the center, there's not a blood type M1/2, it's blood type MN. So no phenotypes are hidden, or nothing is recessive, everything will be expressed. Another example for codominance is roan color in horses. There's a red hair, there's a white hair for the 2 horses. If they breed, the offspring will have both red and white hairs intermixed pretty closely with each other, which will give the appearance of a roan coat. It kind of looks like pinkish. So they're both expressed, there's no blending, except for what it looks like. Multiple alleles, a little more complicated. You have more than just the 2 alleles. For example, our blood grouping system, ABO blood group. Now that I know I can pause, that's going to be nice. So we have A antigen, B antigen, and no antigens. The capital I, which would be the, I don't want to say dominant, but kind of is dominant in this case because this shows both codominance, dominant recessive, and incomplete dominant. I'm sorry, both codominance and dominant and recessive. So we have capital I, which would be the dominant over the little I, A-type antigen, B-type, and none. So what does that look like? If you have blood type A, anybody blood type A out there? I'm blood type A positive myself right here. 2 possible genotypes, I big A, or I big A, little I. Now, if that was a blood type, how could you figure out what your blood type, what your genotype would be if you're blood type A? What kind of cross could we use? Test cross. Hopefully you got that. Blood type B, and also blood type AB. And the last blood type is blood type O. What do you think we're going to have for blood type O? Little I, little I. Again, this would be the law of segregation. When these would put these on a Punnett square, you would have right there in the center. That is 1 allele. All right, another allele, what you traditionally call them, is right there. That is what they would be. The I and the B never separate. That's never going to happen. So this is the diagram from the book. You can check this out, but how the antigens work, how antibodies work, or we can use my little simple diagram. Let's see if this works. There we go. Is it reading backwards? I don't know. Let's see. Oh, you can read it correctly. Good. So these are for blood type. Here's how blood typing works. This is going to be for blood type A. All right. It's going to have little flags on the outside. These are the antigens. Think of antigens as flags, like old armies, Braveheart. They have a flag to say what army they are. These antigens are like the flags. This is also going to have antibodies. Let's go on this side and look at blood type B. And blood type B is also going to have anti-A antibodies. So what happens, these are antibodies that are in there. What would happen with these antibodies is they would come over here, come over there, and they would bind with the antigens and basically cause clumping. So you'd have all, if it was some blood type A blood and the blood type B, all these antigens would start binding these antibodies, or vice versa, and the type A blood would then clump up. And that is why blood transfusions, you have to have the right type of blood. But that's the difference between antibodies and antigens, antigens. Here to RH factor, like I was RH positive. RH positive is dominant, RH negative is recessive. And they would also have the anti-RH antibodies as well. That's another way to separate the blood groups. Epistasis. This is when one gene is going to control a couple of different traits, different locations. The best way to look at this is, can't go backwards, is examples. So we have two genes for color. We have albinos, and you're going to have either brown or black. So in this case, letter C, this is going to express whether you have the color will be expressed or not. Will it have color or will it be an albino? If it's dominant, it's going to have the color. In this case, going to cross a brown with a brown parent. Brown, this would be the black allele, would be the small. Brown allele would be the dominant. The F1, they would all be brown because they'd have the C allele for the color, the dominant, and they would have the B allele for the brown. But when you get to the F2, I'm sorry, when you continue on, in your first generation, 9 will be this. Do the Punnett squares. 3 of them will be black because they'll be either big C, or big C, little c. They're going to have color, but they have little b alleles. So they have 2 black alleles, so they're going to be black. But if you get one that has both the little c and the little c, that's going to be albino. It doesn't matter if this is big B over here or if it's going to be little b's over here, because this is the recessive. This gene is saying, I don't care what signal you're going to have, you're not going to show alleles. It's not going to happen. You're not going to show the color. So that is an example of epistasis. So what's going to happen in that, you're going to have hidden traits come out. All of a sudden, where does albino come from? Ratios are not going to be 9 to 3 to 3 to 1 or 3 to 1. Ratios are going to be off. That is explained. Polygenic inheritance is when you have many genes affecting a trait. So if you remember hitchhiker's thumb, you have the gene for it, or you don't, or you have the gene for hitchhiker's thumb, or you don't. It's either or. There's only one gene that controls it. Polygenic. Poly means many. So you have many genes that control it. And usually it's going to be a gradual curve. Change in phenotypes. Think of hair color. Think of height, human height, skin color. So the genetic basis. You're going to have several genes. For skin color, we don't have one gene controlling skin color. That is why you can have parents. One parent is lighter, one parent is darker. You can have kids in between, a kid with darker skin or a kid with lighter skin. Or the same thing for height. If you can have dad is 6 foot tall, mom is 5 foot tall, the kids can be 4'5", they can be 5'5", they can be 7'5". That'd be a good basketball player. So it's not going to be, because it's controlled by many genes, there's many different phenotypes as well. Diagram from the book, one of the book even has more steps than this one. But you can see, think of these dots. Think of you have 3 genes controlling it. So here's your 3 genes. Each parent would be heterozygous, 1 dark and 1 light allele. So they're both going to be similar skin color, 3 dark and 3 light alleles. Now, what if one kid got all 3 of these light and all 3 of these light alleles? They would have 6 light alleles, very light skin. What if another kid, let's say, got this dark one, but these two light alleles, and ditto over here. One dark and these two light, because it doesn't matter, it's law of segregation, get any of those you can. You would have 1, 2 dark, and 4 light alleles. So you'd be ending right here. So it's polygenic. You see the bell curve. We've heard a lot about the bell curve with coronavirus recently. All right, this is your bell curve, a nice gradual change. So a kid can be lighter than both parents, darker than both parents, or even the same, or somewhere in between. That's when you have different traits around the families. Again, the smooth flow of the traits, the characteristics. Genetic studies in humans, I think we're going to do this one, the next one. Yep, we're going to make a new video because we're about a little over 10 minutes, and we'll be back for part 3.